Good morning. I'm Yufeng Zhao from Xian Medical University in China. I'm very pleased to present my recent work. My presentation is GPR120 the regulation of immune and endocrine function. Free fatty acids are not only the metabolite for energy supply but also involved in the metabolic regulation in cells. With the discovery of plasma membrane receptor for free fatty acid, another aspect of free fatty acid in regulating cell function emerged. Free fatty acid acts as extracellular ligand to activate G protein-coupled receptors and the following intracellular signal transduction in many kinds of cells. There are at least 4 different membrane receptors for free fatty acid, and we focused on GPR120 to clarify its physiological and pathophysiological function. GPR120 knockout mice fed high-fat diet exhibit more severe obesity phenotype than wild-type mice, and GPR120 agonists increase fat loss and improve insulin sensitivity in wild-type mice. It is suggested that GPR120 is the target of drug development for metabolic diseases. GPR120 is present in taste bud, white and brown adipocytes, enteroendocrine cells, and pancreatic state cells and macrophages. It has been reported that GPR120 is present in enteroendocrine L cell, K cell, S cell, and the other cells. GPR120 activation increases the secretion of glucagon-like peptide 1 and GIP, CTK, and neurotensin. It inhibits the degradation of ghrelin and somatostatin. Different signaling pathways are proposed to mediate the effect of GPR120 activation on enteroendocrine hormone secretion. In macrophage, GPR120 activation inhibits pro-inflammatory cytokine release, including tumor necrosis factor alpha and interleukin-1. Multiple signaling pathways for GPR120-induced cytokine expression have been provided. In our study, we aim to clarify the role of GPR120 in regulating endocrine function and immune function. Here we presented our work about the role of GPR120 in pancreatic polypeptide cells and in alveolar macrophage. For the identification of and purification of GPR120 expressing cells, GPR120 EGFP mice were generated. The sequence of raised EGFP was inserted in the genome behind the gene terminating codon of GPR120 by CRISPR-Cas9 technology to generate the knock-in mice. The mice express EGFP under the control of GPR120 expression, and GPR120 expressing cells can be identified by EGFP fluorescence at 488 nanometer excitation. In contrast to the wild-type mice, the GPR120 expressing cells in colon and adipose tissue can be observed clearly. The isolated islet of EGFP knock-in mice showed fluorescence. It demonstrates the expression of GPR120 in islets. To clarify the cell types of GPR120 positive cells in islets, the islet of the EGFP knock-in mice were digested to single cells for enrichment of EGFP positive cells using flow cytometry. Since the expression of EGFP and GPR120 were measured by RT-qPCR, we found that EGFP and GPR120 EGFP positive sorted cells were significantly higher than that in unsorted and negative sorted cells. It indicates the successful separation of GPR120 positive cells from the mixed islet cells. The EGFP positive cells have significantly higher expression of somatostatin and pancreatic polypeptide, and low expression of insulin and glucagon. In accordance, EGFP negative cells had significantly lower expression of pancreatic polypeptide, somatostatin, but higher expression of glucagon and insulin. It indicates that GPR120 is present in PP cells and somatostatin cells. To further verify GPR120 present in islet cells, single-cell PCR was performed using single-cell EGFP-positive cells. RNA was obtained from single EGFP-positive cell, and the expression of GPR120, pancreatic polypeptide, somatostatin, insulin, and glucagon were measured by nested PCR. Of 12 cells that were measured expressed GPR120, and in these cells, 8 cells expressed PP and 4 cells expressed somatostatin. None of these cells expressed insulin and glucagon. This observation further confirmed that mouse islet PP cells express GPR120. Immunocytochemical staining was performed to demonstrate GPR120 protein in PP cells. It shows that the co-localization of GPR120 and PP in mouse islet cells. The role of GPR120 in PP secretion was then investigated in vivo and in vitro. Peritoneal injection of GPR120 agonist TUG891 for 50 minutes significantly increased PP levels in mice compared with the control with vehicle. TUG891 incubation of mouse islet for 50 minutes also significantly increased PP levels in medium compared with control. Islets from the knock-in mice showed same response to TUG891 as islets of the wild-type mice did. The increase in intracellular calcium concentration was known to mediate GPR120-stimulated GLP-1 secretion. We then observed whether intracellular calcium is involved in GPR120 activation-induced PP secretion. Here, GPR120 expressing cells of the islet of the knock-in mice were purified and cultured for intracellular calcium concentration measurement. PP cells were further confirmed by post-recording immunocytochemical staining, and their change in intracellular calcium concentration were analyzed by recording of the intensity of calcium indicator XROT1. The PP cells showed stable level of calcium level during the recording and the treatment of vehicle. In contrast, the cells exhibit significant increase in intracellular calcium concentration by TUG891 stimulation, and the intracellular calcium concentration returned to the control level after removal of TUG891. GQ protein inhibitor YM254890 and phospholipase C inhibition by U73122 and the depletion of intracellular calcium stores by subdigagon eliminated TUG891 stimulated the increase in intracellular calcium concentration in PP cells. GQ protein inhibition, phospholipase C inhibition, and subdigagon treatment also significantly eliminated TUG891 stimulated PP secretion in cultured mouse islets. The effect of GPR120 activation on PP secretion was then observed in high-fat diet-induced obese mice. High-fat diet mice showed increase in body weight and fat mass. Unexpectedly, we found that the islet of high-fat diet mice had a significant decrease in GPR120 expression compared with the control non-control lean mice. The control mice showed a significant increase in serum PP after TUG891 stimulation. In contrast, also high-fat diet mice showed an increase in serum PP by TUG891 treatment. the level of increase was significantly abated than that in control mice. The islet from the control mice responded well to secret PP after TLG891 stimulation, but the islet from high-fat diet mice showed a significantly reduced PP secretion compared with that from the control mice. This study demonstrates that GPR120 is expressed in mouse islet PP cells and is functional to stimulate PP secretion. GPR120 stimulated mouse PP secretion is mediated by GQ protein, phospholipase C, and intracellular calcium signaling pathway. The stimulation of PP secretion by GPR120 activation in high-fat diet-induced obese mice was significantly diminished. It is proposed that the decrease in GPR120 activation during high-fat mice may facilitate the occurrence of obesity. Another part of work about GPR120 is its role in macrophage motility and phagocytosis. It is known that GPR120 activation inhibits the expression of pro-inflammatory cytokines. Macrophages not only release cytokines but also process the action of movement and phagocytosis. We see observed the effect of GPR120 activation on movement and phagocytosis of mouse alveolar macrophages. Mouse alveolar macrophages continue to move actively throughout the recording process in multiple modes, including pseudopods, stretching, and migration. After being stimulated by TLG891, the macrophages immediately tend to become round and exhibited a significant reduction in the movement, including pseudopod stretching and migration. We next observe the change in intracellular calcium in macrophage using flow-3 dye. Flow-3 fluorescence intensity remains stable at basal levels throughout the recording process in macrophages in placebo control. In contrast, macrophages that were treated by TLG891 showed a quick increase in fluorescence intensity, which remained high during TLG891 incubation and fell down after removal of TLG891. GQ protein inhibition, phospholipase C inhibition, and subcutaneous treatment all significantly suppressed TLG891 stimulated increase in intracellular calcium in alveolar macrophages. GQ protein inhibition, phospholipase C inhibition, and subcutaneous treatment suppress the inhibitory effect of TLG891 on macrophage movement. Chelation of cytosolic calcium by BAPTA also significantly suppress TLG891 inhibits the movement of macrophage. Mouse alveolar macrophage in placebo control swallows the fluorescent microspheres and showed increased fluorescence as measured by flow cytometry. In contrast, the macrophages treated by TLG891 had decreased fluorescence. GQ protein inhibition, phospholipase C inhibition, subcutaneous, and BAPTK significantly suppress the inhibitory effect of TLG891 on phagocytosis of the macrophage. In summary, GPR120 activation increased intracellular calcium in mouse alveolar macrophage and inhibits their movement and phagocytosis. It indicates that TLG891 inhibits the motility and phagocytosis of mouse alveolar macrophage through GPR120 activation and the GQ protein coupled phospholipase C and the intracellular calcium signaling pathway. The molecular mechanism of the inhibiting of macrophage motility after an intense increase in intracellular calcium are not clear. It is proposed that inappropriate local fluctuation of intracellular calcium in a certain pattern ensures the normal movement and phagocytosis of the macrophage. The intense rising of intracellular calcium that was stimulated by TH891 led to a continuous strong contraction of skeletal protein in macrophage, which caused their inability of free stretching and consequently restrained macrophage movement. In summary, we presented the role of GPR120 in pancreatic polypeptide cells and alveolar macrophage. Thank you for attending the meeting. Thank you.